So thanks again for everyone who's signed up for the webinar. Thank you for those who are joining us. Good morning, good afternoon, wherever you might be. We're excited to start talking to you today about busting cancer patient recruitment bottlenecks. Hopefully everyone on the call is aware of the problem. We will go through some of that today. And I'm very happy to have with me today a couple of colleagues that we've been working with and that have a lot of experience in this space. So just to start off, I'm TJ Bowen. I am the Chief Scientific Officer and co-founder at Deeplens. I'll tell you a little bit about Deeplens, but we focus on trying to help solve problems in recruiting patients for oncology clinical trials. And I'm joined today by Deborah Friedman, who's a Director of Clinical Research at Hoag Hospital in Southern California. Deb has an accomplished research career with 30 years experience in clinical trials. She has spanned a complete spectrum of pharmaceutical sponsors and clinical research organizations, working with physician practices and hospital-based programs. She has a passion for leadership. She has clinical research operations and managing high-performance teams. She has successfully managed over 600 adult and pediatric inpatient, outpatient trials for phases 1 through 4 in a multitude of specialties. And so thanks, Deb, for joining us today. I really appreciate having you on.
Thanks for having me, TJ.
I'd also like to introduce Jim Langford. Jim is the Vice President of Clinical Operations at iVita Biomedical. Jim has been in the industry for over 25 years. He's founded and managed several companies to leadership positions. He was the founder and president of The Phoenix, which was a CRO that provided clinical research services to the biomedical industry. That organization, Jim also founded an electronic data capture software company, Data Labs, which was spun out and eventually acquired by Parexel. 2009, he formed a consulting company where he provides executive management services within several gene and cell therapy companies to assist them improving clinical development processes. So Jim, thanks to you for being with us today as well.
No problem. Thanks to be here.
All right. So, sorry, a little bit about us, about Deeplens. Deeplens is a startup company based in Columbus, Ohio. We are focused on, as I said, clinical trial pre-screening and recruitment of oncology patients. And what we do is we differentiate ourselves because we try to focus as early as possible in recruiting those patients. We start at the time of diagnosis, and we follow patients throughout their journey, tracking changes that happen and tracking differences in their progression of disease. We surface that information to providers in order to help them make better decisions and help their patients get on the therapy they'd like to get on. We have a lot of experience in oncology. We've worked with providers and pharma and have a lot of AI experience. Our engineering team is very deep in the healthcare space, understanding data analytics and integrations into hospital systems. We have acquired and developed our IP in this space around AI and genomic ingestion as well as search. And we have a little bit of a unique business model that we'll talk about a little bit more in detail later. But the first thing I wanted to do is talk about what we're trying to get across today. I think it's important to understand the key challenges in recruiting patients. So we're going to spend a little bit of time talking about general challenges that are being faced, and then more specifically asking Deb and Jim to kind of elaborate on some of the challenges they see related to those issues. We're also going to talk a little bit about how technology can help free up resources within the healthcare community so that those folks can be more productive and find patients that are slipping through the cracks. And then also learning how to automate and identify screening processes and how those things can help provide real powerful insights to both providers and sponsors of studies. So first topic, key challenges in recruiting patients for these precision medicine studies. I'd like to start by just throwing up some pretty staggering statistics. A lot of you are probably familiar with these, but it is an extremely expensive endeavor when you take into account all the sunk costs in getting a drug to market. The pharmaceutical industry spends billions of dollars per drug to get these drugs to market. And for every day that that drug is not launched, it actually costs them a lot of money just in time that that drug is not on the market. The bigger challenge, however, I think that's more important is that the patients that are really wanting to get on these cutting edge therapies are not able to get on them. Part of the challenge that we're really focused on today is the clinical trial part of that process. And you can see some statistics here that are really indicative of the larger problem. 25% of clinical trials fail to fully enroll. Almost half of trials are delayed due to patient recruitment specifically. And I think the most important statistic is that there are a lot of patients that would like to be on clinical programs at some point during their journey, and only 5% of those that are qualified actually ever enroll. So these are some big problems, and I think Jim is probably the best positioned to speak a little bit to this. And I'll just ask you, Jim, when you see numbers like this, what really jumps out at you and why?
Well, there's two things, TJ. The first one is just, it's on the left, $8 million a day. When I started in the CRO business in the late 80s, that was under $1 million a day. So the cost of doing clinical trials has gotten appreciably higher. And a lot of that does have to do with recruitment. The fact of the matter is that we just can't find the number of patients. I'm not surprised by the seeing that only less than half meet the goals that we had set for enrolling in clinical trials. What was shocking, though, was the 5% of qualified patients, because when we think about doing clinical trials and I'm a sponsor, I'm going out to the clinical sites that I know of, that I'm aware of, or through the CRO brings us in contact with. And these are people that do research on a regular basis. So if you think about it, though, what percentage of hospitals or clinics or clinical settings actually do clinical trials? And it's a relatively small number if you look at the overall healthcare paradigm. So this is not a surprising number, but it's obviously one that we're really trying to see how can we get out and find more patients that would participate in a clinical trial, which would allow us to meet our goals on time, which would really reduce the cost of doing clinical trials.
Yeah, I don't disagree with that. And Deb, I'm going to put you on the spot a little bit and just ask if any of these numbers, particularly on the right, the percentages, are any of those surprising to you, given what you see in your role in managing trials?
No, I wouldn't say the numbers are surprising. I mean, that's really running the average of what we see in trials. And it's very disappointing because not being able to reach full enrollment actually delays the entire process for getting things approved.
Right. And as we're talking about this, I think just an illustration of what's really happening here, there's a lot of steps involved in getting a drug through the clinical trial process. And obviously it starts with trial design and understanding what the studies need to look like. But I think one of the big bottlenecks is really this identification and screening of those patients. There's a lot, as I mentioned, there are a lot of patients when the polls go out, 70% of patients have a positive view of the clinical trials now. And I think, if you'd ask Jim and Deb, I'm sure that statistic has changed a lot over the past, 10 or 15 years. And I think a lot of it has to do with precision medicine and just the perception that there are these game-changing drugs that are now coming to market that can really save lives. It's really changing the conversation, especially in oncology, from extending life to saving life. So, I think the patient perception is really changing. The challenge is the system is still struggling to keep up with the demand and really trying to find those patients, because as we know, precision medicines are very helpful to the patients that they serve, but it requires more triage to find patients that actually will qualify for those precision therapies. So, you have to know molecular genetics, you have to know diagnostics, you have to know a lot more detail about each of those patients in order to do that successfully. And then as a result of that, there's a lot of people within the healthcare practice that have to be involved throughout this patient journey in order to find the information needed to treat those patients effectively. It starts with the pathologist. As I mentioned, this is where Deeplens really starts our process, is with pathology. A lot of people don't generally think about who does the initial diagnosis. It's actually not the oncologist, it's the pathologist in a lot of cases. So when we first interact with a hospital system, we go directly to pathology to understand the nature of the tumors, the nature of the cancer, the stage grade. A lot of the information actually starts there. There's also obviously the oncologist and the surgical oncologist. There's clinical research coordinators whose jobs it is to make sure that those patients who are deciding to go into a research path get the care they need, they meet all of their requirements, and they follow the protocols that are assessed. And then you, of course, have the principal investigators, and these are the people who are responsible for making sure those trials are successful, the oncologists who are looking into that. Deb, I'm going to ask you a question about this. So as we've all experienced, navigating the health system and as a patient is extremely complex, but could it, it could even be more challenging and potentially for someone who's potentially newly diagnosed cancer patient. Through our findings at Deeplins, we've actually found that care team members vary from one institution for the next. In other words, you might have a nurse navigator or a CRC at one site, but you only have, referring oncologists at another. I guess I'd like for you to tell us about the way your care team looks and how you function in terms of making sure those patients get on the right therapies in the trials programs at your institution.
Yeah, so here at Hoag, we actually centralize all of our research. So everything funnels through our research organization. We don't, we work at multiple locations, but everything comes out of one central location. So all the patients are funneled through that. But I wanted to also include that, you brought up nurse navigators, and they're very important in this process. But I think we're also missing, so here at Hoag, we add in our geneticists and our genetic scientists, especially for precision medicine trials. So they're a part of the mix in addition to those four that we involve in the process of looking at patients for screening for precision medicine trials.
Yeah, that's a great point, Deb. I'd actually like to ask a follow-up question on that. I think that's important. I think that from what we've seen, that is not extremely common. We think that Hoag is taking really strong steps towards that. How have you seen that help at Hoag in terms of having people on staff that are actually experts in genetics? How is that helping your process?
Well, it actually helps a whole lot. So, I mean, as most people know, clinical research coordinators are, jack of all trades, but then in addition to having to understand genetics and all of the different mutations specifically and how they relate and what they mean for a particular trial, it's really, really difficult. So having somebody dedicated who can weed that out for them helps. So we're able to take patients that, potentially qualify for a trial, present them to our genetics team, and have them do an initial review to make sure that the mutations are the correct ones that we're looking for, and then bring it back to the study coordinator to finish doing advanced screening.
Thank you. That's very helpful. I'd like to dive into a little bit more detail about some of the challenges that we see that really face us in recruiting patients, especially for clinical trials in oncology. I think the first big bucket here is that there's massive and complex data that has to be sorted through. If you really think about the way these trials work, a site might have dozens or hundreds of ongoing trials, and each of those trials has dozens or hundreds of patients, which result in thousands of patient lives that are being covered. You then layer on the complexity that each one of those different studies might have its own unique inclusion and exclusion criteria that the people who are trying to recruit patients for have to be intimately familiar with. They have to know ahead of time which specific criteria are going to qualify or disqualify a patient for a study. And then, of course, you have starts, stops, and pauses of each of those studies. So you not only have the data problem, but you have the complexity of having to know when a trial is actually recruiting. In some cases, we actually have sites that have remote study locations. So you might centralize a lot of the process, but you might have satellite facilities that are helping to recruit patients. And then, of course, patients continuously come in. They're always being diagnosed, and staying on top of the number of patients is really hard. These things really contribute to this narrow window of opportunity. As a patient comes in and is diagnosed, they might not initially qualify for a study, but after a period of time, they might not be responding to treatment. So at that point, they might become ready for a study. So all of these things add up to things that have to be tracked and monitored. I think another challenge that we see is limited connectivity and interoperability. So in a lot of cases, we still see that electronic medical records and laboratory information systems aren't always integrated. They're starting to get there, but they're not. We see a lot of different third-party genomic test results. So even at a same hospital, you can have multiple vendors being used by different physicians to retrieve genetic results. And then, of course, when all of that comes together, a lot of times you have it sitting in unstructured data. So that's really hard to sort through when you're looking for a needle in a haystack. And then the last thing that we kind of bucket it under is the ineffective manual tools. And this, from our experience, is an interesting challenge, because what you have is you have all of these sophisticated pre-screening requirements that are having to be managed by people, yet the tools available to them prior to enrollment of a patient are pretty minimal. They're using a lot of times spreadsheets to track patients. We see sticky notes on screens, on monitors. We see, yellow notepads being written on. They're often, copying static EMR pages. They're really trying their best with tools that are not keeping up with demand. They aren't able to communicate as effectively as they need to between the team members. So if a clinical research coordinator has a question for a PI, it's often challenging to get an immediate reply back about the specific question they have. So I guess with all of those things, I want to ask both of my panelists, Deb and Jim, which of these things do you see as being particularly challenging, or do you have any kind of insights into some of these, or any experience seeing any of these? I guess I'll start with you, Deb, from the practice perspective. Which of these do you see as particularly challenging?
Yeah, so, to be honest with you, it all is a challenge. And I want to add one more. One of the issues that we tend to see is that coordinators are extremely busy. And to properly screen a patient can take hours. And when you're screening multiples, tens, hundreds of patients through a system, you're going to have lots and lots of patients who are not going to qualify. And coordinators develop kind of a frustration level because they're looking for patients. They want patients to qualify for trials. And the more that they spend time looking for patients that don't qualify, they tend to not want to screen so many patients anymore. And so I think that that's a cycle that we see also. They're not getting good candidates sent to them to be screened. And so because of that, lots of the patients aren't qualifying, and then they just don't want to screen anymore. So I find that I have to try and keep motivating the team to continue to screen patients for trials. So I think that's a really important factor that's not brought up here, where coordinators just frankly get disappointed.
That's a great point. And I think the other, the thing that adds to that also, that I'm sure you would agree with, is this isn't their only job, right? A lot of times they're taking that patient all the way through enrollment and even into the trial. So this is just a fraction of what they have to be responsible for. Is that right?
Yeah, absolutely. And once a patient's on trial, a coordinator's priority is going to be treating that patient, not looking for new patients.
Right.
So looking for those new patients kind of drops down, and again, kind of funnels into that cycle where they experience disappointment when they're not finding patients who actually qualify for trials.
Right. Jim, from the other side of the coin, on the sponsor side, or someone who's trying to run trials, which of these things resonates from your perspective?
Well, what Deb said is the thing that we always focus on, is that, well, how much percentage of time can a coordinator have in pre-screening for qualified patients? So how much of the time are can possibly be spent? You have to understand, it's a competitive marketplace. I'm sure at Hoag, they're running hundreds of clinical trials. They may run several clinical trials where patients could potentially be in my study or another study. And so you have to really understand that this is a competitive marketplace. There's limited resources for the people to be able to identify. So you have to try and set some realistic goals on what can you effectively expect to get out of a clinical site. This is particularly challenging when you're trying to identify sites who would qualify patients to participate in the clinical trial. How do you know? How do they know how many patients would actually qualify based upon where they are in their clinical plan? Where are they in the process of how they're going to be cared? What is the number of what studies are available to them? And how do you know which patients will actually qualify? It's really a crapshoot. I mean, I really sympathize with the clinical sites trying to come up with an idea of, okay, we can do your study. How many patients can we contribute to your study in a reasonable period of time? Without having any really technology to help them go through and identify what potential patients may look for, they're trying to do their best to say, I think it's about this number, but until we really get into it, we really don't know. And
Right.
so one of the things we really focus on is how much time can a coordinator spend doing pre-screening, and what tools do they have to help them identify patients that, as Deb said, are patients who will actually qualify. So we try to kind of narrow down our inclusion exclusion criteria for screening process to say, here's the three things that really matter.
Yeah.
Look at
No,
those first. If they don't
that
hit
makes
those
a lot of
first three
sense.
things, then move them on. Go on, then leave them out. We'll try and get to the next bucket of patients.
That makes complete sense. That kind of leads us into this next slide, which is really about the
It does.
complexity of trial design, right? I mean, I'm assuming that when you select sites, a lot of it has to do with the expertise available at the sites you're looking for and the ability to find those patients that are very, very specific. I think the complexity of trial designs has really changed. And I'd love to get Deb's kind of input on how it's changed, and you too, Jim, over the course of your careers. But well, I mentioned very early in this conversation that the introduction of precision medicine therapeutics is really a benefit to society, but it's a real challenge for people trying to run studies, because you now have these complex trial designs like basket studies, right? Where historically, if you were a lung cancer doctor, you would know the lung cancer treatments, and probably the lung cancer trials that are available. But now that there's basket studies, that kind of throws that traditional paradigm out the window, because you now have to know all of the molecular markers, and the patient needs to be diagnosed by molecular marker, not necessarily tissue, in order to qualify for a specific study. So that complicates it. I think, the training of oncologists and the rest of the care team is a challenge. I know many oncologists that I've spoken with that would admit that they aren't trained geneticists, they're oncologists. And then when you start throwing in all of this complexity, it just adds more to the plate, and in terms of being able to digest all of this information and provide a patient who's desperate for care the right decision. And then, of course, you then have to imagine all of the different diagnostic tests that you have to discern between which one to use for each patient and when. So I'm going to start with you, Jim, and just ask, when you're designing a trial and trying to find specific sites or PIs to run those trials, how much do you think about these things in terms of, your ability to get that site to actually find patients for your study?
We think about it constantly. I mean, design of study and feasibility have to go hand in hand. There's no point in designing a clinical trial that gives you a really great scientific answer if, in fact, you can't enroll any patients because of the design of the study, or the population you're looking for is so narrow that if you expect a site to get one patient a quarter, or how do you keep that in the forefront of their mind that that's the patient they're looking for? So you have to look at these things at the same time. You have to understand what that patient population might look like. So what's the population that you might see in the country? Then try and narrow it down into actually what's the population you would see in a region versus the population then at a clinical site. So then when we talk to the clinical sites, we really want to talk to them about what's your experience in doing clinical trials that are similar to this. Tell us about the experience that you have, and try to understand and talk with them. I mean, when we do feasibility studies, I think they're somewhat different. Our feasibility studies that we do with clinical sites are not on paper. We don't send surveys or things. We do it in person. We do it over the phone. We have conversations. We want to talk to the physicians. We want to talk to the coordinators about their care plan and how they would treat patients, and think about what type of patient would actually fit into their care plan and the population that they have, so that we can get very specific to say, okay, this patient would fit, this one won't. These would be excluded, these will be included. And then try and get a feel for how many patients do you think you would have, and what's preventing you from getting more patients? Because if there's something that's preventing you from getting the population we need, and it's a secondary or tertiary criteria that we're looking at, we may modify that to make the protocol fit the population to get the primary endpoint resolved in the period of time that we're hoping to do.
That makes a lot of sense. Deb, I'm going to ask you a related question. How have you seen this kind of change the ability for physicians to recruit patients now that everything's moving toward, especially in oncology, everything's moving towards precision? Is this a challenge? How are they mitigating that challenge? Is it something that you guys are working to overcome?
Yeah, so it definitely presents a challenge. So being that we're a community-based hospital, we have numerous community-based physicians who may or may not be even aware of some of the research trials that are ongoing in the community. And one of the things that we've done to try and help bridge that gap is we have a director of precision medicine. So all of our patients who are, let's say, have metastatic cancer, all receive automatic reflex testing on their specimens.
Mm-hmm.
And those results are run through a system, electronic system. They then come to our precision medicine director, who actually reaches out to each individual community physician whose patient this is, to discuss with them about potential treatments, whether it be research or non-research, in trying to help our community doctors to better understand precision medicine and what's best for their patients.
That's great. I'm glad to hear that they're trying to fix that problem. I think a lot of institutions are struggling with this challenge. And again, I just wanted to kind of illustrate one of the things I talked about earlier, which is a different challenge, which is this narrow windows of opportunity to enroll patients. And this was a slide that we worked with Dr. Strickler at Duke University, who sees this problem a lot. And it's really a multifaceted issue where you know patients that come in, particularly for a second line or a third line treatment, a therapy that they're trying to get on a clinical trial for, have to have obviously failed prior lines of therapy. Well, what we find a lot is that the oncologist is only going to wait a limited amount of time before they start looking for that next therapy for their patient. And even if a patient is identified very early in their cancer diagnosis, while they're in their first line treatment, the person who's trying to track those patients down has to remember to come back and check on that patient again after they've already undergone their first line of therapy. And they have a very narrow window to do that. Not only that, they have to be tracking which trials are available. And as I said, some of these trials start and stop based on enrollment. So they might have been tracking a patient towards one treatment, but then that treatment might have stopped for a period of time. So they missed that window of opportunity. I guess, Jim and Deb, with that being said, I'll start with Deb on this one. With some of the numbers I touched on earlier with, the patients that, the trials that never reach full enrollment, the delays that are occurring, the few number of patients that actually get enrolled, how does this narrow window of opportunity play into that? Is this something that you see as being a big contributor, or is this just something that we see from our perspective?
No, I think it's actually a big contributor. So, depending on which kind of protocols you're working on, we run a phase 1 cancer unit here. It's very important for us to catch these patients early. So as everybody knows, phase 1 research used to be for, basically patients who've been treated with everything under the kitchen sink and we're still not responding. Well, phase 1 research has completely changed and it's no longer like that. A lot of patients, even for first-line treatment, are now going on to phase 1 trials. So having the ability to catch those patients early is very, very beneficial and something that we struggle with.
Yeah. Jim, that same question, I know that in some of your even current studies, you run into this issue just based on timing and some of the procedural things that have to happen. I was wondering if you could just touch briefly on, some of the windowing that's important in enrolling patients in some of your studies and how, surgeries and other things can really kind of become important in making sure that you're aware when patients are being given different procedures.
Right. The windows are important because it's where the patient is in their care plan. And so, like we said, like one of the examples is we're doing on an ovarian cancer clinical trial where we need to have tumor from the patients to make our final product. Well, that means that you have to have a window exactly of when is that patient going to have debulking surgery, or they can have a laparoscopy where we can get enough tissue that we can start making the product. And so doing the timing, and then what is their time to their chemotherapy? That is their standard of care that they would be doing. So it may be that they will do neoadjuvant therapy, which means they do chemotherapy for 3 months, they do debulking, they do it for 3 months more, or they may wait 6 months. The timing becomes a variable. And we simply don't know when that window of opportunity will show that this patient will actually have the surgery that's necessary for them to qualify to participate in our study. So it's a challenge for the clinical sites to remember where is that patient? Is that patient in a place that is now eligible for our trial, or are they going to just continue with their therapy or go to a different clinical trial? It's a challenge for us to be able to find that exact window. And this is even for studies where we're not trying to identify biomarkers on those patients. You add
Right.
that into it, where you have to have a patient who has certain response to various treatments. I can't imagine how challenging that can be.
Yep. I think that leads to kind of these statistics, right? These are some statistics from papers that have been published about the time that it actually takes for clinical research coordinators or nurses to actually spend finding these patients. They spend anywhere from 1 to 4 hours a day to 11 to 12 hours per eligible patient to get them enrolled. I mean, these are staggering statistics that you have to spend that much time, and then you try to think about how many patients they're looking at and how many studies they're looking at. I think it goes to say that there needs to be some help. Another interesting component here is that, as Deb has mentioned, in the community setting, we see a lot of ramp up in the community setting. There's a lot of desire to get patients who are being treated in the community onto studies. We know that the vast majority of patients that are oncology patients are actually treated in the community setting, up to 80%, some statistics say. So solving this problem is even more challenging in the community because, as we talked about, sometimes these patients are coming from disparate sources. They're not centralized like they would be at a comprehensive cancer center. They're seeing patients from all over their community. So that's a huge challenge. Recruitment opportunities are really siloed as well. So there's challenges across the workflow. What we see is, pathology has challenges in that they're not necessarily always connected to the care team. Sometimes they just get a sample thrown over the wall and then they send it back over the wall, but there's not really a direct communication in a lot of cases. It keeps them isolated. The focus is really on the diagnostic workflow and not on improving the treatment options. And there's folks that are trying to change that, and I think Hoag is one of those places that's trying to change that. Another issue we see is in the genomics testing. As this becomes more and more prevalent, it's really important that those test results are timely, get turned around in a timely fashion, so that those patients get presented with all of the opportunities they possibly have. As Deb said, I think at their institution, it's great. They're doing reflex testing automatically, where the pathology team doesn't have to wait for the oncologist to send in another request. As they see it come through, they can trigger those tests to be done automatically, which is great because it really reduces that turnaround time. The other challenge we have, though, is when you get those reports back, it actually puts a pretty big burden on the care team. They have to interpret these genetic results. And as we said, there's still undereducation from across the care team spectrum on who's on what the knowledge base is for those things. And then for clinical trial support, we see that a lot of times it's disorganized. There's a really strong focus on the last mile, which means after a patient's gotten consented to get them enrolled, but there hasn't been a lot on those first opportunities. And then connecting the care team is really important. So with that, I wanted to transition into the second part of our conversation, which is really around technology. Just at a high level, we see obviously technology can enhance a lot of these solutions. We can fix a lot of the processes that are currently not working well. We can maintain sort of visibility for the care team and the sponsor. So a person in potentially Deb's role can see what's going on a very granular level, and a person in Jim's role on the sponsor side can get some of that data that he's frankly not been able to see very well because it's just not been captured. And then we're able to enhance the real-time communication across sites. I think I would like to, because we've been obviously worked with Deb and Jim in the past, I'd like to ask each of them, how implementing technology is really helping some of your problems. And Deb, I think focusing for you on just kind of, reducing staff burden, like what have you seen or what do you see as the potential for technology to help with that problem?
Well, really what's happened is due to technology is able to identify better quality candidates for us to screen, so that my team doesn't spend hours and hours and hours screening patients who never really should have been screened in the first place. Mm-hmm. So technology is narrowing down the sample size for them to look at, but is providing a better quality patient for them to actually screen. So it is actually improving our numbers for adding additional patients to trials.
Great.
And I think Jim, that it's,
oh, sorry.
I was just going to say, I mean, the most important thing, especially right now in the midst of COVID, is resources. And anything that we can do to eliminate wasted time is extremely beneficial right now.
Absolutely. I mean, we're seeing sites really struggling with COVID to do anything but COVID. So, the cancer patients are the ones who are suffering. So that's unfortunate and something that we think we can help with. Jim, from your perspective, on the sponsor side of the coin, how do you see technology on the provider side helping you?
Well, it's what I'm excited about is the fact that it gives me and the clinical site a platform of which to have a common communication. Traditionally, it's if enrollment is behind at a clinical site, what are my options except to call the site and say, why are you behind in your enrollment? And whatever a response they give me is the response that it is. There's nothing that says that we have the ability to have an objective conversation regarding, well, what patients were actually screened or could have been screened. So if you have the ability to have technology that'll give us a common platform that we can look at basically the same information to say, there were 7 patients that we pre-screened and only one of them were able to get enrolled in the study out of those 7, and let's talk about the 6 that maybe weren't, or why weren't they? And you have a conversation that isn't just one-sided. It's a two-sided conversation talking about an enrollment, and it opens up a level of communication that you just generally don't have in communicating with your sites these days.
Yep. Yep. We're excited to be a part of that process. And I think, the Viper application is really trying to help find some of that information that's been missing, frankly. And it's not the fault of the provider and it's not the fault of the sponsor. It's just data that hasn't been captured. It's really nice to see that happening. Really quickly, I just wanted to touch on this. This is just kind of a view of the screening process, and I think what we've seen is traditionally the orange in this slide is really where a lot of the technology has been focused. It's really been focused on that last mile of once a patient's enrolled, they then get enrolled into the EDC system and their information starts getting tracked. They then, start working with clinical trial management systems that track every step of the way. But there's this whole series of steps that happen before that where there's not really a lot of energy being put into the technology component. Deb, I would like to ask you, you've been using Viper at your institution for several months now. How could a solution like Viper help healthcare institutions streamline this process and bust through these bottlenecks that we've been talking about?
Yeah, so I mean, what we've seen here at Hoag is that with the ability to use Viper, like I said previously, it's identifying better quality patients. So in the initial process of setting up the trials within the Viper system, the inclusion-exclusion criteria that we feel is the most important is what's selected, and then we'll go through the electronic systems to pull these patients out of the system to notify the coordinator that they have patients ready to be screened and for which particular trial they need to be screened. So that's been very effective because, like I said, it's giving us a better candidate to screen versus a physician just saying, here's a patient, take a look at him. Right. Or them going to a tumor board or, one of those other facilities and trying to do all of the work themselves. So they're able to more centralize all of the data that they need to be able to properly screen a patient. So I think technology has, great advantages. It has to be used correctly. Right. It has to be implemented correctly. Everybody needs to be properly trained, but it streamlines the process because then the coordinator can go straight to whoever the provider is and discuss with them and let them know that they have a qualified candidate and then start to move through that process.
Yep. And to that point, one of the things that we try to do with our solution is really help the community providers. As you said, there's a lot of information that's flowing through the system, and there's a huge strain on the people that are involved. And one of the things that we really focus on is trying to process a lot of that upfront information for you so that you can focus on the patients who are more fully qualified. We also realize, though, that there is not ever going to be a perfect AI solution for this. There's too much nuance to it. So what we try to do is capture the data that will help those CRCs or nurses or whoever the care team member is that's in charge of finally getting those patients consented and enrolled so that they don't have to look at, all of the patients who are never going to possibly qualify, but just focusing on those that are close to qualification. So, we have a lot of systems in place that automatically sort that. We integrate with those lab systems and with the EMRs to triage out patients who are not going to qualify. But we also keep the ones who could potentially qualify in the future in the queue, so that they'll continue to pop up if that's something that's going to potentially work for them. I think another thing that we do is we help identify potentially previously missed patients. So this is a challenge. We know that there are patients that are being missed. I don't think you could go to any hospital in the United States and get an honest person to tell you that they're not missing any patients. They cannot order molecular tests on every patient, first of all, due to the cost and lack of confidence in reimbursement. But even a lot of times patients who should be getting tested are not. We know that a lot of lung cancer patients, NSCLC patients, are not getting tested who should be, even though NCCN guidelines say that they can be. So it's an education issue. It's a patients falling through the crack issue. One of the things that Viper does is actually identifies patients who most closely match those trials and precision therapies and reduce that risk. And then we send recommendations to pathologists so that they can get those tests ordered if we think they haven't been ordered correctly. Another thing that I just wanted to touch on is the way that data is processed. So traditionally, one of the huge challenges in this space has been, as I mentioned earlier, the disparate data sources. You get unstructured notes fields, you get feeds and messages in the system, you get reports that are PDF'd, you get inputted data from the care team members, and these all sit in their original form in the EMR, which require either a person or really smart systems to go through it and process that. One of the things that Deeplens has spent a lot of time on, and we have a lot of experts that have done this in the past in their lives working on, is really integrating all of these disparate data sources into a single data-centric or patient-centric data model. We use machine learning, we use natural language processing, and we use fielded data. And we really try to create a normalized and controlled set of data so that can be easily searched within fields in the system. Deb, I want to ask you really quickly, there's just so much information coming from so many different places. I have to imagine your CRCs are overwhelmed. I guess, can you talk a little bit about, you mentioned it earlier, but a little bit more about how, they've gone more to a referral model just because this is so intense and so much time it takes to get the pre-screening done.
Yeah, so, we kind of have a different model here at Hoag, being that we have, a director of precision medicine and we have genetic scientists, which a lot of facilities don't. So we have a lot of assistance for some of our doctors. For our coordinators, though, being that majority of coordinators are having to become getting very familiar with technology, it's really proven to be quite a benefit for us. We've been working with you guys now for quite some time, and we've actually seen patients that we've missed. We know that we're missing patients, and by being able to enable technology into our workflow, it's helping us to catch the things that we've missed or to save time from the things that we used to spend hours and hours and hours working on. So technology for us has been fantastic. We have a very large EMR system here, which is not so easy to navigate when you're searching for very particular criteria. A lot of physicians don't document in the correct place, or a lot of physicians use outside sources, and then it goes into a different section of the EMR and makes it very difficult to find. So being able to have all of the information in one place and having one company help us pull all of our systems together and all of our data together just saves numerous amounts of time. And really, time is of the essence.
Yeah, no, we've appreciated that as well, and it's been really helpful to work with a group that's doing those things, because we learn from you too. Really quickly, this is kind of a general workflow that the Deeplin software uses to help with that, as Deb was mentioning. What we do is we start with sponsored trial data. So we work with the vendor, with the sponsor, the pharmaceutical company, and with the site to really work on which studies they want to load. And so we can work on the protocol directly from the sponsor. We can work off of data that's publicly available, but we really curate that content and extract out all the inclusion exclusion criteria into discrete operating fields. We also integrate with the EMR and the LIS systems, as well as some of the molecular genetics tools. And what we do is we pull data in from those systems automatically. We use algorithms and AI that we've built that will parse out the relevant information and really put it into those fields. And as that happens, we match that against those clinical trial criteria that we have pulled out of those studies. When that happens, based on specific criteria that the care team has said they would like us to triage, we can then notify them and send them an alert saying, hey, we have a few potentially qualified patients that you should look at. And what they can do then is come in and use the workflow that we've created for them that will show them the patient, it'll pull out the data, and it'll help them really navigate to the critical information they need to do, saving them a lot of time in getting those patients either qualified or disqualified. And it also keeps a lot of that data, so we understand why a patient's being not chosen to be consented or why they're not qualifying. That information is valuable because it can be presented back to Deb, so she can see, you know, hey, we're seeing a lot of patients that might have the molecular marker, but they're not having a specific other attribute. Maybe they haven't failed the right type of prior therapy. Maybe that's not something our physicians typically do in our protocol. So that information is very helpful. We present that data back through a dashboard so that they can log in and see how many patients there are in the funnel, how many patients have qualified or been disqualified along the way. And then what that does is it really lets them further that process so that they can get that complete screening done and then get those patients enrolled as quickly as possible. I'd like to just show sort of some of the data that we worked on with both Jim and Deb over the past several months. We introduced Viper to them at the end of last year, I believe, and you know, we started it out by just saying, hey, let's do an experiment and look and see what we can find. And we chose 20 different studies that Hoag was running, you know, we looked at these studies, and what we did is we implemented Viper using a single CRC who was untrained. She had never used the system before. We asked her to do the pre-screening for these 20 studies. She picked it up very quickly. She spent a few weeks going over months and months of retrospective data, and she triaged, you know, a lot of these patients down to about 150 potential patients, as Deb mentioned, that were really unidentified before. And it wasn't because they necessarily weren't qualified. It was just because, you know, there is a strain on the system. So the other complexity here was that these patients weren't just homogeneous patients. This was across 16 different types of studies we found patients in, had 12 different biomarkers, 11 different tumor types. 3 of them were multi-arm basket studies. So this was a complex analysis that was done. I think this was just kind of proof that with a system, you can use a single person to do the job of multiple people historically that are frankly spending their time doing other things as well. So I think, Deb, I would love to just end it by letting you kind of make a last statement, you know, what you've seen in terms of the changes that you've seen at your institution, what you hope to see moving forward, and then I'll ask you the same question, Jim.
Well, I think this is still a work in process, and one of the things that's been so fantastic about working with you at Deeplens is the fact that you listen. So you listen to what's working for us and what's not working for us, and you have the ability to modify that to make it be as effective as necessary to work with our site or work with any other site. You're able to, you know, personalize it to what we specifically need to look for our trials. Not everybody is doing the same type of trials, even though it could be precision medicine. It might be a phase 3 precision medicine trial, which will have very different requirements than working in a phase 1 trial. And so that's really one of the greatest things for us. We've loved our relationship with you guys and are looking forward to continuing that relationship and moving forward in the future and being able to roll this out to as many trials as possible.
Great. Thank you, Deb. Appreciate that. We feel the same way. I think it's been great. We've learned a lot. You know, one of the things that we've learned, I'll just throw in there, is that our model has evolved to really include providing sites with the CRC. So, you know, we don't just throw software over the fence and say, you guys figure it out. we've evolved our solution to the point where we're providing actual people with boots on the ground to help get these things across the line. We know that the teams are burdened. We know that it's hard to throw them just yet another tool and another task to do. So it makes it much more efficient for us to provide someone on site to really kind of help with that process in whatever way they need us to help. And that's something we've learned by working with you guys, so we appreciate that. Jim, I'll ask you the same question. What have you seen from kind of working with us and just working with technology, and how is it changing the ability to potentially run trials moving forward?
Well, it's TJ, you know that I'm impressed with the results that we've had from the pilots that we've done with Hogue. I mean, for the audience, I'm one of those studies, and we actually were able, through that a 3-month period, identify 9 patients that could have potentially been in my clinical study. And as it turned out, 2 of them were still qualified because we had a window that was wide enough to allow that patient to still get into the study. So we've actually seen direct results from being able to talk to clinical sites regarding the use of this technology to help them identify patients for the clinical trials. So much so that we've gone out as a sponsor. I've gone to all my other clinical sites that are doing these studies and encouraged them to look at Deeplens and Viper. We've had conversations regarding this has worked in some of the sites that we've worked with. You should look at this. We're not in a position that we're going to, as a sponsor, where we would mandate the use of software like this, but we will encourage our clinical sites going forward. You should be looking at this type of technology because it is meaningful. It impacts the way that we do our clinical studies.
Thanks, Jim. Appreciate that. We have just a few minutes for question and answer. I do see that there's a question in the queue. Let me see if I can pull it up and read it. So, to improve treatment over diagnostic, what is the potential of small therapeutics and matching patients to more innovative precision therapeutic opportunities? I think that's really what we are talking about is, these potential small therapeutics. Jim, I would say, you're well qualified to answer that question. As a biotech as opposed to a large pharma. Can you maybe try to address that question about how it's helping with more innovative precision therapeutics?
Well, yeah, because we're looking at, because we have so many new criteria, because we're doing besides just precision, we're actually doing personalized medicine. And so we're looking at very specific patients to which we will be treating with a personalized therapy. And so for us, identifying the right patient and making sure that patient can actually qualify both from a personalized point of view, that they can actually do the genetics, we can have all the testing that's necessary in the timeline as necessary, is critical. And we're not looking at lots and lots of patients. So you're hopeful that you can get, I mean, seriously, 1 patient a month is a fabulous enrollment for most of the studies that we're doing with these very complex studies. It also is meaningful in the fact that our studies are becoming so much more complex. We're trying to answer so many more questions with a single protocol because the cost of studies are so much higher. So it means that you have to be looking at every patient who is possible to get into your study, that you can actually do a meaningful screening and find the patients, and stretching out into the community outside just the research centers to find the patients that can participate in your clinical trial. It's extremely competitive at this point in time.
Yeah, we've definitely seen that. That's great insight, Jim. I think we're at the end of our time. I really wanted to thank both Jim and Deb. It's a great partnership we have with you, and we really appreciate your insights, and hopefully the audience appreciates it as well. I wanted to end by just saying, if anybody has any questions or would like more insights, please reach out to us at Deeplens. We're happy to talk to you more in detail about our offerings, or even about some of the the work we've done and how we're seeing results. So again, thanks to both of our panelists. We really appreciate your time. Thanks for everybody who's attended, and we look forward to speaking to you in the future. So thank you, Jim, and thank you, Deb.
Thank you, TJ.
Thank you for having us.
All right, have a great day, everyone. Thanks again for attending.
Bye-bye.
Bye.